Hello students, in continuation to the cell and its components, I am going to talk to you about nucleus and its chromatin material, which is divided into euchromatin and heterochromatin and nucleolus. I am Dr. Uma Masur, Assistant Professor, Pavithubai Chokle College of Arts and Science, Margao. Outline of this session is chromatin and its molecular organization, nucleosome, DNA packaging in eukaryotes, DNA, histones and non-histone proteins, nucleolus and ribosome structure in brief. At the end of this session, you will be able to describe structure of chromatin and its molecular organization, identify nucleosome, explain DNA packaging in eukaryotes, discuss DNA, histones and non-histone proteins, as well as describe nucleolus. Let us look at this chromatin and its molecular organization. As you have studied the nucleus, you know very well that the nucleus appears in its prominent form during the interphase stage. When at this stage the cell is stained with basic fission, there will be certain things which will be stained and certain things which will be not stained. With this, we can understand the chemical nature of chromatin. Chemically, this nucleus will be having the presence of DNA and RNA. Predominantly, it will have DNA and RNA only 5%. Along with this DNA and RNA, it will also have proteins which are histone and non-histone type. When you analyze the content quantitatively, it was found that protein and DNA exist in the ratio of 1:1. That means, along with DNA, proteins are also important for its structure. It is also found that histones are present in eukaryotes and they are absent in fungi. Depending upon the stain taken by the chromatic material, we can differentiate between two types of chromatin, that is heterochromatin and euchromatin. Heterochromatin is darkly stained, condensed and is deposited at periphery of the nucleolus. It is metabolically and genetically inert with less amount of DNA and high amount of RNA. Euchromatin is stained very lightly. It is in a diffused form with high amount of DNA. That itself tells that euchromatin is nothing but the true chromatin. When we are studying about the molecular organization of chromatin material, we have to understand that chromatin is suspended into the nucleoplasm during its interphase stage. As the cell cycle follows, this nucleus will also show some changes. You will find that nuclear membrane is disappearing and the chromatin material is becoming more and more complex and dominant, as this fibrillar structure will become more and more condensed and shorter, giving rise to the full-fledged chromosomes. There are lots of sequences which are taking place from the movement of chromatin material to the formation of chromosomes. And one of the important stages is nucleosome formation. What is this nucleosome? The DNA, as we have studied, exists with the proteins in the ratio of 1:1. So this DNA is a tightly bound mass of histones. Nucleosome model was presented by R.D. Kornberg in 1974, and according to him, it is made up of octamore of core histones. Which are these core histones? They are H2A, H2B, H3 and H4. These octamer of core histones are present in multiples of 2 and they form core of DNA. If you look at the diameter of this nucleosome, it is 11 nanometer and the height of this nucleosome is 5.7 nanometer. Double-stranded DNA helix is found to be wound around this nucleosome, which ranges in the base pair of 146 to 200. There is also a linker DNA which is about 54 base pair long. It starts bonding around the octamer histones and it ends with the linker DNA. Now this linker DNA is sealed off on this octamer histone with the help of histone H1. Now let us look at the DNA packaging process, which is also called as the solenoid model of packaging. As I have informed to you, the chromatin material will follow the sequence of organization, levels of organization from a very thin DNA double-stranded helix, which is 2 nanometer, to the formation of a proper chromosomes, which is about 1.4 nanometer. The sequence of level of organization which happens during this period is very complex. It starts with the prophase and ends with metaphase. As we begin, the chromosome is 2 nanometer wide, showing a DNA double helix structure. When this short region of DNA double helix further condenses to 11 nanometer, you will find that they are going around the nucleosome. This stage gives the appearance of beads on the string. The H1 histone, which are sealing of these DNAs giving rise to the formation of nucleosome, are actually in communication with each other, and that is how they come together and further become compact. This continues to give rise to 13 nanometer wide complex, wherein the chromatin fiber is further packed into nucleosomes, giving rise to 13 nanometer fibers. It is observed that the 6 nucleosomes come together and form a tube-like structure as they go on coming together, and the scaffold-like structure is formed inside this structure. Further condensation takes place wherein the chromosomes are found to be in this extended form and ranging in between 300 nanometer diameter. At this stage, the chromatin is 40 times condensed. Later, you will find that as it reaches to metaphase stage, it is 1.4 nanometer in diameter. At this stage, when the histone depleted enzymes were used, it gives the appearance of a scaffold-like structure with a halo made up of loops of DNA, and it was also found that non-histone proteins are further folded to 100 times over here, and that is why the more compact form of chromatin is seen at the metaphase stage. Metaphase chromosome is the stage wherein the highest level of nucleosome organization is seen. As a component of the nucleus, DNA is also important part. As you know, DNA is deoxyribose nucleic acid. It is the main genetic constituent of a cell, and it carries all the genetic information from one generation to next generation. It exists in complex with proteins called chromatin. Now let us look at the histone and non-histone parts of the nucleosome. Histone proteins are basic proteins, and they are with a high amount of arginine and lysine types of amino acids. Because of the presence of these amino acids, which are basic, they bind tightly to DNA, which is acidic in nature. There are different types of histones like H1, H2A, H2B, H3 and H4. Histone H1 is lysine rich histone. Histone H2A and H2B are slightly lysine rich. Histone H3 and H4 are arginine rich histones. H1 is involved in higher order foldings of chromatin. Core histones are involved in holding together DNA molecules in chromatin fibers. Non-histone proteins are diverse in nature. The number of these histones also vary depending upon the stage of the cell and the species, and they seems to be regulating the specific genes. They form the 50% of the structural proteins, which are supposed to be made up of actin, alpha and beta tubulin, and myosin. They show active participation during cell division. Nucleolus. In its nucleus, the presence of nucleolus. There may be one or more prominent spherical, colloidal, or acidophilic nucleolus. There seems to be absent in bacteria, fungi, sperm cells, muscle cells. They are predominantly found in oocytes and secretory cells. If you look at the location, they are positioned at eccentric side of the nucleus. They are often associated with nucleolar organizer. If you look at its chemical composition, you will see that this is not a cell organelle. It does not have any limiting membrane. The amount of calcium ion is higher over here, which maintains the intact organization of nucleolus. It contains DNA of nuclear organizer, ribosomal RNA, ribosomal proteins, RNA binding proteins, etc. Nucleus has a very important function to perform. It makes ribosomal subunits from proteins and ribosomal RNA. It sends the subunits for the complete formation of ribosomes from the nucleoplasm to the cytoplasm. Ribosomes make proteins, and therefore the nucleolus plays a vital role in making proteins in the cell. Different stages of ribosome formation takes place in 3 different regions of the nucleolus, starting from center to periphery through the stages of initiation, production, and maturation. It goes through the stages of fibrillar center, dense fibrillar component, and granular component. Nucleolus keeps in touch with the cycle of nucleolus and the cell. It disappears at prophase, metaphase, and reappears after telophase, that is, when the cell division has taken place and 2 daughter cells are reaching to its maturity. The references used for these presentations are Verma and Agarwal, Gerald K. and Patil C.R. Additional references www.biologydiscussion.com. Thank you.